Hey guys, so this video is going to just give you an overview of the very first plasma membrane lecture, specifically going over just the structure and composition. Now, probably a lot of you maybe have had this before in your undergrad classes, but you've probably never had it along with this much material before at this fast a pace. So hopefully this video can be sort of an island of sanity for you while we try and figure this stuff out. So without further ado, let's get started. So there's going to be three different common types of membrane lipids. We have phospholipids, we have glycolipids, and we have cholesterol. And this is about as good as my handwriting is going to get, so you'll just have to bear with me. Now, membrane lipids have an amphipathic nature, as we have here. Amphipathic just means that they both have hydrophilic and hydrophobic parts to the molecule itself. So hydrophilic, as most of you probably know, prefers an aqueous environment, while hydrophobic would prefer a non-aqueous, which is fairly apparent if you have any grasp on Latin at all. Now, hydrophilic and hydrophobic also go by the name of polar and nonpolar. With hydrophilic being polar and hydrophobic being nonpolar. And now the hydrophilic part of any membrane lipid is the head group, and the hydrophobic is called the tail group. So now we're just going to look at the structure of a phospholipid because those are the majority of the membrane. Now a phospholipid has four different parts actually, even though this only has three colors. This is an alcohol over here, and this is actually a phosphate group here. And this phosphate and alcohol make up the hydrophilic head group, or the polar group. And so, like I said, there's four groups. This gray backbone here is glycerol. And these two green boxes are fatty acids. Now it's important that you actually get this structure down, because this is something that they will test on, just knowing that the basic structure of a phospholipid has two fatty acids. I'll write in the word acid. And a glycerol backbone with a phosphate and alcohol head group. So a few other important takeaways that now that we have the structure down is that these fatty acids can be unsaturated or saturated, which you've probably heard about before. Now saturated just means that it has hydrogen connected to all of the carbons and it's fully saturated with hydrogens. There's no double bonds. If a fatty acid is unsaturated, that means there are double bonds. And usually what this means is that there's a sort of a kink in the chain. So normally a fatty acid chain will just be sort of a up and down chain like this. But if you have a double bond in there, you'll get something like that, which will just put a kink in the chain. And then this is actually really important when it comes to the structure of membranes, because if you have kinks in the chains that make up the inner part of the membrane, these parts in here, like this would be the outside of the membrane, and this is the inside. So the inner parts here, if you have a kink, that actually makes the membrane more fluid. So if you have a lot of saturated fatty acids, it's a very rigid membrane, while a lot of unsaturated fatty acids will make it a very fluid membrane. And now going along with this discussion of rigidity of membranes, like we mentioned earlier, that a major type of membrane lipid is also cholesterol. Cholesterol acts as sort of a buffer. So if you have a very rigid membrane, adding cholesterol will actually make it more fluid because it breaks up the sort of the monotony of the fatty acids lined up. And if you have a very fluid membrane, cholesterol actually acts to sort of pack things together and make it more rigid. So it can play both roles, so they refer to it as a buffer. And the reason that we care so much about membrane rigidity is that it's actually inherently tied with melting temperature. So a really rigid membrane, a really rigid membrane will have a very high melting temperature, while if it's a very fluid membrane, it'll have a much lower melting temperature. And even on top of this still, if you have a very, very long fatty acid chain as opposed to a very short one within the membrane, a very long chain will actually be easier to come apart. It'll be more fluid, so it will have a lower melting temperature, whereas a very short chain will actually have a higher melting temperature and will be more rigid. And if you want an analogy, just think of Legos. Like a very long block of Legos is actually really easy to break up, but you get down to like 2 or 3 Legos, you can never break them apart. So now we can just talk about what kind of stuff can get through a membrane. Now, we have here just different categories of stuff that can get through the membrane or can't. So they refer to this concept as selective permeability because some things can permeate the membrane, some things can't, so it sort of selects what it can. So really small things, like and they have to be uncharged. They can get through the membrane, and gases can get through the membranes, and they can just pass right through without any resistance whatsoever. Now, water and urea here are both unique in that they can get through, but it depends on the bulk flow. So it just depends on the concentration of water outside the cell and how much osmotic pressure is there to push the water into the cell. Otherwise, if the cell is under a lot of pressure itself, the water can't get in. That being said, so these are the molecules that can get through, and it's important to have these down and understand the concepts behind these. Now, the things that can't get through are large polar molecules, so like glucose here is given as the example, and it's just too big to get through the membrane. And you also have ions, of course, because they're charged, so they can't get past the membrane. And then finally, charged polar molecules, which is just combination of being charged and being a polar molecule, then those also can't get through. And these are also usually going to be large. Now, I say this, they can get through the membrane, but they simply just require an active process like a channel or a pump or any other sort of complicated protein structure that's going to sort of actively get the whatever material you're trying to get into the cell. So then, now that I've mentioned channels and the like, we can move on to what other stuff that we can find inside the membrane layer. So channels and pumps are going to be proteins that are going to be integral within the cell membrane layer. But they're also proteins that are peripheral to the cell membrane layer, so they're just sort of attached to it. So they don't actually pass through the hydrophobic aspect of the membrane on the phospholipids. They just sit on the outside near the polar end, and that's the peripheral proteins. But they're also the integral ones that I've previously mentioned, and these will be channels and stuff like that that actually pass through both the polar and nonpolar aspects of the membrane and sit within it. And now, this is really interesting because you can remove peripheral proteins fairly easily. You just have a salt or pH concentration different from what the cell likes, and then they'll just be washed off. But integral proteins are extremely hard to crystallize and sort of isolate to study because they're within this membrane. So you have to use really harsh conditions to get them out of the membrane, but then you destroy the protein in the process. And now, along with this, you can also have proteins. That actually are they are connected peripherally, but the connection that they have is to another protein or molecule that sits within the membrane. So it's sort of anchored within the membrane. It's different from peripheral because peripheral just sort of sits on the edge of the membrane. Well, this is going to be directly bonded to a molecule that sits within this membrane. And they'll generally refer to those as anchors. And so you'll just have, say you'll have a molecule here that sits within the membrane, and then you'll have a protein that's attached to that anchor. So it's sort of in between the peripheral and integral. So now that we've sort of looked at proteins on a large macro scale, we can look at them as sort of individual amino acids and how they break down within the cell membrane. So usually, like I've mentioned, you have a very hydrophobic area within the cell membrane, and then it's hydrophilic on the outsides. And that's exactly what this figure shows. You have a bunch of these yellow amino acids that are actually hydrophobic. So usually they're very packed together, but this doesn't necessarily mean that you have to have all hydrophobic amino acids within this cell membrane. So you can have a couple of these charged or non-charged, or even the polar ones within this, as long as sort of the majority of them are going to be hydrophobic. And so the very final point to make about this figure is, it's very good at showing this, is the sort of the asymmetric nature of the protein on either side of the cell membrane. So you have the inside of the cell here and the outside of the cell here, and you have these green diamonds here that are actually sugars, they're glycoproteins. And these are really important when it comes to cell signaling. So these glycoproteins are added to the protein in the ER and Golgi, and so you'll have this protein formed, ready to be placed into the membrane, and then it'll be glycosylated afterwards and have these, well, not green diamonds in real life, but they'll have these glycoproteins added to them. And then this, and like I said, it's extremely important for cell signaling because, and for instance, so the specificity for blood groups, blood types, those antigens will recognize the patterns of these glycoproteins and then react accordingly. So if you have the wrong sequence of glycoproteins or the wrong configuration, then you won't be able to accept a certain blood type. So in summary then, when we're looking at the plasma membrane, these are the things we want to focus on. Plasma membrane is mostly phospholipids. Phospholipids have 2 fatty acid chains, 1 glycerol backbone, and then an alcohol and phosphate group, the alcohol attached to the end of the phosphate group. Now, the membrane itself ends up with a polar head and a nonpolar tail, and that ends up as the configuration of the associations of nonpolar wanting to be together and polar wanting to be near aqueous environments. And now, the membrane themselves can also have cholesterol and glycolipids. Cholesterol acts as a buffer, which both helps the membrane either stay less rigid or more fluid. Less rigid as in more fluid, or more rigid when it's too fluid. Now, the fatty acid chain that I mentioned earlier, if they are very long, then they will have a very low melting temperature, and they will be very fluid. If they are very short, then they will have a higher melting temperature. If they are unsaturated, that means they will be more fluid, and that when they are unsaturated, that means that they've got a double bond going on somewhere. Now going back to composition, the proteins within the membrane can either be integral or peripheral, and then there's also anchor proteins, which are sort of in between peripheral and integral. Finally, it's important to remember that the membrane itself, once it's entirely composed together, is going to be selectively permeable. So certain substances like gases and small uncharged polar molecules can get through very easily. Water can, it depends on the osmotic pressure, and then charged ions and charged polar molecules aren't going to be able to get through without some sort of assistance from a channel. And that covers this lecture, the first plasma membrane lecture. I hope it helps you guys, and my name is John Lukowski, and I wish you guys way more than luck.